Yep.
Awesome. So today I figured that something that would be an interesting topic of discussion for our group, because it's something that I've been thinking about a lot, is whether, I've seen the past few papers that have come out on engineering neuromuscular actuators and tissues from the bottom up, which is the theme of this month's series. Have kind of picked either top-down or bottom-up approaches. And I kind of see advantages and disadvantages to either of those techniques and feel that they've been able to accomplish different things. And I was wondering whether we as a community should think about, is it worth, have we gotten to the point where we should maybe just pick a lane and all focus our efforts on the same thing, or if we think that there's an advantage to it's going both ways. I will put the caveat that I have not included examples here from the CAM lab because I thought that Claire would cover them. So that's the difference. So first, I wanted to motivate why I think we still do need a multicellular approach for our actuator technologies. The state of the art with just muscle is kind of where I left off in my PhD, right? We can get these muscles to twitch with on-off control, either with electricity or light stimuli, and they can move in any direction we tell them to, and that's great. But I think something that we've come to realize as a community is that that alone isn't enough, right? So the dream, of course, would be to have a tissue, a multicellular machine that sends a lot of different inputs, not just light or electricity, but pressure, chemical signals, pH, whatever it is, process them, prioritize them in some way, and then make some sort of decision about that output response. And my dad sent me this video just yesterday of my mom's garden, and I felt like it was too perfectly timed not to share. But this is an inchworm that is hanging out on one of my mom's plants. And I just thought it was really interesting because even though we would always talk about the inchworm inspired design, it had been a while since I'd actually seen one in the wild doing anything. I thought it was really interesting to see how it's sort of sensing every single part of its environment and then making a decision on like where the surface is and what direction it wants to go. So in this relatively simple natural system, we see this very complicated neural processing of multiple inputs and also multiple degree of freedom response. And it reminded me that we are so far away still from that worm. And made me want to think about this neuromuscular idea a little bit more deeply. So again, I'm going to do sort of a case study of top-down versus bottom-up approaches that we've seen both in our group and also a couple other groups as well, and also talk about things outside of the peripheral nerve skeletal muscle paradigm and by talking about the enteric nervous system a little bit. And then I have a bunch of questions that I thought would be useful for us to discuss. So where we left off when I left the Bashir lab a couple years ago was this paper that Caroline and I published. And this was basically a very simple adaptation to our traditional device design approach. We would make a ring of C2, C12 skeletal muscle tissue, same as before. But after letting it differentiate for a few days, we would add a second layer of this ring-like structure into which we would add these motor neuron embryo bodies. And you can literally see them, right? It's like all of these little black blobs that are in the second layer of tissue. And what we saw is that if we had just one layer of muscle rings, there wasn't really any spontaneous twitch that happened, which makes sense. We weren't telling the muscle to turn on in any way. But when we included the second layer that had these motor neurons, we started seeing spontaneous twitch occur after several days in culture. And we confirmed that the spontaneous twitch was due to the formation, the extension of neurites and the formation of neuromuscular junctions by testing the effect of chemical stimulation. So it's like, oh, we added glutamate to the media. With more glutamate, you were able to excite, higher frequency contractions. When you add curare, a toxin, you cut off the communication and you don't see muscle twitch anymore. So it's like, all right, great. We're seeing that this twitch is because of the added neurons. But the big problem that we faced and it's going to come up again and again in all of the examples that I mentioned, is that we saw localized twitch. These contractions that I'm reporting are really things like this tiny little piece of muscle right there was doing something like this, but we weren't seeing anywhere close to that coordinated twitch response that we saw with the previous biobot examples, because reinnervation wasn't happening in every fiber within that tissue. And I think that that was the really big issue that we faced, and we couldn't get past it by the time that both of us left the lab. Thankfully, we left Onur behind at Illinois, and he published this paper last year where they were able to see a little more coordinated functional response. And I think maybe this is a little bit closer in scale to some of the things in Roger's lab, where because the muscle tissue is maybe a little bit smaller, or at least similarly in size to the neurosphere, or there's some sort of formed guidance or channels, you see neurite extension, neuromuscular junction formation and growth, the muscle twitches, and they were able to get this swimming robot to locomote in one direction that matched simulation. And I think this is a really, really big advance, but you can tell that it is quite slow as a result of the fact that it's a small piece of tissue in a big device, right? So I think we still maybe don't understand for a mesoscale tissue how we can form neurite connections or neuromuscular junctions with every single fiber in a large chunk of muscle. So these are kind of, I think, the bottoms-up approaches that we've tried. They're fairly similar, but depending on matters of scale or how smartly we encourage neurite outgrowth, we can tweak the outcome a little bit. And of course, the most recent paper out of this group is more of this top-down approach. I really should have defined what I mean by bottoms-up and top-down before I started, but essentially when I say bottom-up, I mean when we engineer, every part of it is like a tissue that we engineer from the bottom up using either a cell line or some sort of stem cell-derived source, induced pluripotent or embryonic. Anytime that we are relying a lot on something that already exists, like a primary cell or a primary tissue that we excise, or implanting something in an animal, I would consider that a top-down approach in my book. So this sort of top-down approach that was recently published out of Illinois by Colin Kaufman showed, again, you start with your same baseline of our engineered skeletal muscle tissue, and rather than doing what we were doing before, which is like placing a bunch of motor neurons at different places around the tissue, they just excised a piece of a spinal cord from a rat, I believe, and placed it right on top of the biobot. Again, I don't think they saw coordinated, large-scale twitches or locomotion, but they did see something very interesting, which is that all of those little localized contractions were synchronized with each other, indicating that there was sort of a central pattern generator, which was taking place, which was occurring in this lumbar section of the spinal cord that they had resected. So there is, while we're still struggling in this case with massive large-scale innervation of all of the muscle fibers within the tissue, we do see that when you have this central processing occurring, you see something more similar to what happens maybe in our bodies. And you can start thinking about, if we could solve that innervation problem, it would probably be nice to have this sort of central processing system occurring in a spinal cord like area, because you would be able to, all of the input stimuli could come into one direction and the output could happen in a lot of different directions, rather than stimulating each of several embryoid bodies separately. So I think that was a great advantage with this approach. And now I want to highlight a couple things outside of our EBICS domain before I kick off sort of the questions that I thought would be helpful for us to talk about. So the first of these top-down approaches is out of Lisa Larkin's lab at the University of Michigan. And they do skeletal muscle tissue engineering for volumetric muscle loss. So as many of us know, small damages in skeletal muscle can be pretty readily recovered by our bodies by recruiting satellite muscle cells to the site of damage, proliferating, differentiating into new fibers, and you're good to go. But the critical volume loss, and this varies from animal to animal, but generally between 15 to 30% of volume of the original muscle, if that's lost, you've created a critical size defect where you aren't able to fully recover the force generated by new tissues being added. So this is what they're showing here, VML stands for volumetric muscle loss. They took a biopsy punch and they took a big chunk of tissue, actually it might not even been a biopsy punch, it might have been something bigger, out of sheep. So this is a large animal model. So they take a big chunk of tissue out. You can see that over time, the force does regenerate, but it's significantly lower than the 100% value that it was before. And so what they do is slightly different from our approach. They don't do injection molding. They make a muscle monolayer, let it delaminate, it rolls up into these little layers, and then they make a whole bunch of those. They stack a whole bunch of those together and stick it back inside the sheep. And when they did that, they saw that they were able to recover, not significantly greater force than just the pure muscle loss and native repair, but it was significantly great. It was somewhat greater force generation with this case. But then what they were again realizing that the problem that they face is similar to the problem we face, in that they had that volumetric muscle loss had cut off a portion of the peripheral nerves. I think it was over a centimeter. And it takes a certain amount of time for the nerves to grow back. And there's actually a funny thing. I talked to, over the past year, a lot of orthopedic surgeons, and they all say, nerves grow at about like 1 millimeter a day. And they say this a lot, but I couldn't actually find too many citations to back that up or to verify whether that was like a linear growth or anything like that. This is kind of off topic. It's just something that a lot of surgeons told me, and then they were like, we don't actually know that. It's just something we say to patients to tell them that it'll take a certain amount of time for them to regain control, but just something to think about. So these people are thinking, all right, there's this piece of peripheral nerve that's been cut off and it's taking some time to grow back, and maybe it's not growing back to the right location. So they made these engineered neural conduits, or ENCs. Essentially, it's kind of just a collagen tube. And they wanted to basically tell the resected or the cut neuron, grow through this tube and then go to this piece of muscle that we have assembled and placed. And when they did that, you can see that they were able to recover force almost indistinguishable from the native force pre-damage, and significantly greater than in the no repair case. So now what they've done, right, is like they got the nerves to grow to the right part in the muscle, but they didn't really do anything to tell it to innervate every single fiber that they had engineered. It automatically happened, and it happened because it was happening inside a sheep. And so I'm not sure that we could recreate something like this in the lab by just having a conduit, because obviously we've tried approaches like that before. And I think with this sort of big volume of tissue, we would, based on our previous approaches with bottoms up and mesoscale tissues, I'm not sure that we could do it. So I think there's still an advantage here of incubation in the sheep that gave this sort of huge improvement in functional response. And then the last thing I want to show about this sort of incubator approach is that I think we're very used to thinking about peripheral nerves and skeletal muscle, but I also follow the enteric nervous system and smooth muscle space fairly closely, because it's actually very interesting. It's quite a bit farther behind than we are. I think, we've been very lucky in that for a long time we've understood, you add horse serum to the media, you can get this myoblasts diffuse into myotubes and they contract. And people have been trying to do sort of smooth muscle tissue engineering as well, and getting sort of these like coordinated peristaltic contractions in engineered smooth muscle tissue has been a huge barrier, and not a lot of people, really pretty much nobody has been able to get that kind of contraction unless they got an ex vivo resected tissue and then were using it. But a couple of years ago, some work out of Jim Wells' group in Cincinnati, their children's hospital, made a really big breakthrough in this space, and I thought I would share it with everyone. So here they have these intestinal organoids, so that's this clump of cells on top, and they did a bunch of studies where they just showed, the formation of these intestinal organoids, and were like, oh, look at all of, these different layers of cells that had formed, but they weren't really able to get anything similar to smooth muscle peristaltic wave function in any of the organoids that they had made. And they made some gastric ones, upper gastric, lower gastric, intestinal. They were never really able to get contraction. But then they added in this additional clump of cells of neural crest cells And then they co-cultured them together, saw the formation of things that, it looked like, oh, here are some neurons, there were some neuroid extensions, they found some glial cells, and they saw the intestinal epithelium, but they still didn't see contraction or anything like it. And then they incubated it in vivo in a mouse, I think in like the kidney subcapsular space. I need to double check that. And I think it's worth watching this video because it's really cool. It's a very different kind of contraction than we're used to seeing. It's sort of all the way around the wall, but they started seeing these sort of smooth muscle peristaltic waves propagating. And this is really big in the smooth muscle space to get something like this, and they weren't able to get it. And I touched base with one of the postdocs who worked on their gastric organoids. They haven't really been able to do something like this outside of an animal model yet. So there's still a huge advantage to be able to study these kinds of things, to doing this top-down approach of helping them form these connections. So those are kind of some of the examples that I wanted to highlight. And since the purpose of this is just for us to chat and think about these things, these are the questions that I had reading these papers. How far are we really from bottom-up innervation of mesoscale, like centimeter scale, engineered skeletal muscle? What can some of these top-down approaches teach us about engineering from the bottom up? Should we start thinking about moving beyond just are we missing something? Do we need to vascularize these things first before we start thinking about having neural innervation, or does that increase complication? Should we pick a lane, obviously? And then I would just be curious, if we did have this optimally functional neural controlled muscle actuator, what should we do with it? What should that big paper be? And as I mentioned, I haven't highlighted Roger's work because of Claire's talk, but obviously that can be a part of the discussion as well. So I actually can't tell if, should I end my slides and let Claire talk? I can't tell if she's here because I can't see everybody.
Oh, I don't see her here yet.
Oh, okay. So
I think you should keep the slide. I think it's useful.
Sure.
So, Ritu, great. Thank you for that. I appreciate that. Can you tell us, so in terms of starting the discussion, can you give us some quantification of the innervation? So, both from a spatial perspective and other quantifiable means. So, where is it that we're failing? Is it the density? Is it quality of the connections? Is it the graded connection, so that if it's coming from one side, it's more on one side rather than the other side?
Yeah, I don't know if I can put numbers on it, but I can tell you that my gut is that it's not necessarily the quality of the connections, but the fact that those muscle fibers, at least the ones that we were making, are on the order of like 0.5 millimeter to 1 millimeter thick. And I think we did see some amount of grading, like things that were closer to the motor neuron bodies were obviously the ones that were being we were evoking contraction from. So yeah, I think it's just a question of how do you get neurites to extend further into the tissue, but also direct them in some way such that, I think maybe we made a mistake of placing independent neuronal bodies on different portions of the tissue, and maybe we should have done something that was more central, radiates outward in some way, and also has some sort of feedback to recognize, in the body, we do have muscles that are innervated by multiple nerves. We have that redundancy built in. But I think that's the question of how do we, one, get every single fiber innervated, and then should we think about the fact that many fibers probably need to be redundantly innervated in the course of, in case of damage or disease.
So do you have so just following up on the quantitative aspect, do you have a notion of, are we at 10% of where we should be, or are we 50% of innervation? And
I would say it was less than 10 in the biobots, at least. And maybe that's just because they're very big. I'm sure Rogers are probably much higher.
So a couple of ways to approach that.
Mm-hmm.
It's interesting to compare the force we can generate in these in vitro models to what what you can generate in vivo. And we're still very far from in vivo, I think, in any of the in vitro systems. And I think there are a couple of reasons for that. One certainly is, the innervation issue, like you say. But if you start to get something like a, half millimeter or millimeter of tissue, the vascularization is also going to come up.
Yeah.
And, I think we can't ignore that. With the implants, I think they very quickly become vascularized. So that's why in that one study that you showed, they could get back up to pretty much normal levels of force.
Yeah.
But I think in vitro, unless we find a way to vascularize or keep the tissues down, probably with something as metabolically active as muscle, I would think that, maybe a maximum of 1 to 200 microns would have to stay below in order to have diffusion alone be enough. So I think there are a lot of things that we need to do in vitro before we can get to the level of performance that we see in vivo.
Yeah. But do you think it's worthwhile to test that? Is there a way to test the hypothesis of like, if the vascularization problem was taken care of in vivo, then innervation would happen better in these constructs? I'm not sure if we could do something like that.
Well, I think in vivo, it's hard to separate one from the other because they're both happening simultaneously.
Yeah.
From the few experiments that we've done though in vitro, where we had motor neurons co-cultured with vessels, they do tend to help promote each other. So in one of Tatsuyu's papers, he was able to show that he got better performance from the vascular system by co-culturing with the motor neurons and vice versa. So I think there's a lot of communication that takes place by bidirectional signaling, and that's probably important.
I see that Cara has her hand up.
Yeah, I'm curious. So you talked a lot about, and I get mixed up between top down and bottom up. I think that's bottom up. But I'm wondering if you wanted to reverse that. So we learned a lot from just engineering and kind of forcing the cells together in kind of some controlled spatial patterns. But what if you approached it maybe from an organoid, and how much of what we learned from the engineering approaches can we apply to maybe try and develop these from an organoid perspective?
Yeah, I'm not super familiar with I don't even know if I've seen too many works of like peripheral nerves, skeletal muscle in like organoid form. Have you, Roger? I've seen a lot of cerebral organoid work in peripheral, but not necessarily with skeletal. I'm getting no. I think you're muted.
Yeah, no, I agree. I don't think I've seen any either.
Yeah,
I think it would clusters of motor neurons, but it's not really an organoid system, I think.
Yeah, I think it would be maybe I think it would maybe even be more successful, Kara, just because of the scale issue again. That like maybe we would be able to, if the muscle had some sort of tether, because there isn't that much of it, maybe we would be able to innervate more or most of it, and we would be able to see something similar to how they were able to do with the enteric nervous system. Actually, maybe better, because I think with the enteric nervous system, they still, they don't know how to recreate even a portion of that in vitro, which we do with skeletal muscle. So I think they definitely needed that in vivo culture, but maybe we wouldn't if we tried to do an organoid.
So Ritu, in terms of thinking about kind of the organoid or co-development aspect of it, when is the ideal timing? What's a progression for innervation, relative to developmental stages, relative to, overall mass? Because, imagine this, in the fantasy scenario, I imagine that we would start with iPSCs, and they would, you would get co-development, and they would exactly form at the right times. And so my mind turns to, right away to, would we be able to apply, synbio approaches to ensure that, the initial iPSC population each one of them, you know, subpopulations take different lineage tracks in a way that also would, you know, hopefully minimize requirements on different media, or they would generate kind of their local microenvironments, you know, as needed. And then they would, you know, in development of this multi-organoid system, they would innervate at the right time and maybe continue to innervate as a developmental process.
Yeah, no, I actually think that that's very worth doing, mostly because reflecting back on the conversations that I've been having with a lot of surgeons recently about this work is that I talk to orthopedic surgeons that operate primarily on like adult patients with injuries, but I also talk to a lot of folks at Children's Hospital, for example, where they primarily look at people who have like nerve or muscle injuries like right after birth, and they said that that space is very poorly understood, that if that development baseline twitch doesn't occur right away, that they're not able to fully fix, at least with like brachioplexus injuries and things like that. Even if they see everything come back, they aren't able to fully fix it over the course of this population. So I wonder, and also I know this work, again, I think weirdly, because we're so good at engineering skeletal muscle tissues, maybe that's why the organoid space isn't very well developed, but at least in the intestinal organoid space, a lot of that work is focused on understanding things like Hirschsprung's disease. Hirschsprunger's? I never know how to pronounce that. But it's basically that there's a portion of, I think, your intestine or colon that isn't innervated at birth. And so those people are never able to recover that motility even after all sorts of interventions later on. So I think this sort of approach that you're mentioning would probably be very helpful for understanding, at least in some of the less well understood childhood or prenatal diseases, what we could do or what the stages of innervation need to be. At least with the biobots, it was, I think we would differentiate the muscle in a separate media, and then when it was fully differentiated, add the second layer that was some combination of their nerve media and the muscle media. But I need to double check. I'm assuming, Roger, in your systems, the media is kept separate, but there's some communication through the channels.
Yeah. I mean, obviously the neurites connect, and then the media for the two actually are separate, but there's diffusion, cross-diffusion between the two channels. I was wondering, is there anything to be learned from the cardiac organoids that people? I mean, we're starting to work with a little bit now too. You know, they're just they're grown from IPS. They're just differentiated as a mass into cardiomyocytes. And then they beat on a regular basis without any innervation at all.
Yeah, I mean, I guess the question that, well, with those, and I guess even with the intestinal to a certain extent, there's sort of that pacemaker, but the pacemaker still needs to activate everything. But I guess there, because the gap junctions take control of propagating that signal, one thing that we could learn is, I guess we could think about, as Rashid has always mentioned, like, you know, can we engineer gap junctions or some things into these muscular systems, which would maybe work for the bots. Like maybe if we don't want to solve the innervation problem and we just want to solve propagation, we could just engineer the gap junctions and learn from cardiac organoids that way. It probably wouldn't work for, I mean, it's definitely not going to work for any of the sort of more medical reverse engineering applications of these kinds of
technologies. That's true. Yeah. But
Well,
Michael
I don't
has
know
done
if
a lot of
people
work.
want us to try that. That seems like a lot to get approved.
Yeah, Mike Levin has done some interesting work putting gap junctions into even epithelial cells so that they can communicate.
Oh.
So that might be an interesting approach to try.
I was curious when you mentioned the role of vascular cells and having the signaling. Do you think it's just biochemical signaling between the three different types, or is there some role that mechanical twitch or expansion or pressure is also playing?
With the interactions between motor neurons and the vasculature, I think that's probably mostly biochemical, because there really isn't. I mean, there's nothing's contracting there. But I think when you start talking about muscle, I think the contraction, it has to have an impact, because the cells are going to be deformed. They're going to be responding to that stress. And I don't know, I suspect it would be a response that would tend to favor vascularization and maybe favor innervation, but I'm just guessing.
Okay, that makes sense. Actually, one thing that I'm working on right now in the lab is, the effect of exercise on innervation. And I think it would be interesting to see if we had the motor neuron tissue and the muscle tissue separate versus sitting right on top of each other, whether it makes a difference, because that way you would be able to kind of separate those variables of biochemical communication and mechanical.
I guess one other experiment that Tetsuya did was to have a muscle strip between two vessels that were vascularized and had endothelial monolayers. And there he looked at the effect of exercise on angiogenic sprouting. So clearly there's something. The factors that are being secreted by the muscle, due to the hypoxia, I think probably are inducing the vascularization.
Yeah, I found sort of preliminary results still is, so for those of you who don't know, I developed this tool for interstitial fluid sampling from the brain, but I thought that it could be very useful in some of our 3D tissues, because it allows for, generally when you try to sample these biochemical signals in the surrounding media, they're at such low concentration that it's like hard to tell. So I've been sticking these sort of very microinvasive probes in different parts of tissues that I engineer. Some of the tissues actually from Li Hui Sai's group, Roger. And we've seen that we can see much higher concentrations of signaling chemicals and proteins if you sample from within the tissue rather than the surrounding media. Where was I going with this? Oh yeah, so where I was going with this is I think that could maybe help us answer some of these questions of like how muscle acts as a paracrine organ on either surrounding vasculature or with nerves, and maybe kind of simmer down, like if we could even, maybe you don't need to exercise in the future if we can figure out what those factors are, and if we can deliver that in some other way, that could also help with the innervation. I guess one thing I was curious about is, I think my obvious take, if we fully understood this innervation problem, I think one great place to start would be in vivo with volumetric muscle loss, just because in that case, I mean, they were able to recover a lot of the force production, but if you actually look at, histologically stain it, there's still like a ton of damage and the muscle is kind of a little bit malformed. So I think that would be a great place to start if we at least knew where to place the neural conduits in a smarter way that we could encourage full reinnervation. But I don't know if other people have thoughts on other impactful areas or experiments we could try.
When you compare that with and without the sheep, or even why were sheep chosen for that?
Because it's a large animal, so that was mostly it. They wanted to show that they could scale up to nearly human size with their like roll-up model. So it was more of like a manufacturing challenge for them. They had done it in rats before.
And you get the same general results, just smaller?
Pretty much. It's just harder to make that volume of tissue. So they've done a pretty good job with it.
And if you don't, I mean, if you do use the best, like in vitro media and whatever, what do you get from that? It's like as a negative control.
With neurons or without, just?
With the whole system, but rather than putting it in sheep, just do the negative control in vitro.
Gotcha. I don't know if they've tried it, but my guess would be that they probably wouldn't be able to innervate even like 5% of that muscle, just based on our experience with much smaller tissue, and theirs is like a big chunk. I think it would be like night and day compared to what happened when they placed it in situ. And
so in terms of the, either the topology or the architecture that you got there, is it spatially distinct, is especially separated from the other sheep internals, and it's just that there's signaling that's being provided, or there's a realignment of the cells as well within that in vivo environment?
I think for the most part, because they knew the alignment of the tubes, it was pretty physically obvious. I can try to pull up actually that paper, and maybe it'd be useful to, let me see if I can do that. I have them open. I have a preprint version, so it is not the prettiest thing, but I can show some of the pictures. Preprints are the worst. All right. Okay, so you can see that that's like a big volume of tissue, right, that they're putting back in there. Real big chunk, looks like 15 centimeters or so. So again, this is like orders of magnitude bigger than anything that we've done, and it looks like they resected about 30%. And then 1 centimeter was the area that nerve had to grow to get to the engineered muscle tissue. So it wasn't super long. I mean, 1 centimeter is, if we follow the 1 millimeter a day approach, it's not crazy long, but who knows if that's actually true and how soon it occurs after damage. This is some of the in vitro characterization. So you can see like the fusing and nice alignment of the cells. And I think they place it along the axis of the alignment when they implant it. So it probably, you don't see too much change there. And then what is this? Gosh. Oh yeah, so these are their in the collagen just channels that they put in. There's nothing special there. I mean, one thing I do wonder is if we did something like this, but instead of collagen did something that was like a smarter coating inside that encouraged neurite extension, might also be helpful. And then this is all just data I showed before. I'm trying to see if there was more of that staining data that would help answer your question, Juan, about alignment. It's here somewhere. Okay, so yeah, so this shows at least a little bit of what the damage and staining looks like. You can see that there's a ton of fibrosis, even in the best case healed tissue scenario. Not great, but at least it gives you a sense of like, yes, there's muscle, it looks like it's aligned. I'm not sure that we actually see where the engineered neural conduit is in this case, so I don't know if we can answer your question, but you do see that there's like a ton of shenanigans that shouldn't be there. And maybe if they left it in there and let it remodel over time, you would see it go down. But it's also worth looking into. I wish we could see where the ENC was placed, though. I don't think that that's really very clear in this paper. Oh, here, a little bit. Yeah. I don't think there's too many other exciting pictures now. It's just this. But yeah, this is made me sad because I was very excited. I thought they'd solved it all, but I think with that amount of fibrosis, you'll see an issue over time.
So do you think there's a lot of cell migration that's either beneficial or damaging within that context? Cell migration to the locale?
Yeah, I think probably they're not able to turn off that we're damaged approach as much. Maybe they should be putting something in addition to just, you know, muscle cells and collagen. I wonder if there's something they could put in their engineered construct to like tamp down the immune response. But I also wonder if they haven't optimized the stiffness of their scaffold as much as they could, and maybe they're directing pro-scarring, pro-fibroblastic lineage in that tissue. That's actually something I'm working on with a cardiac fibrosis project right now. Like rheologically characterizing tissue as you like change the softness and see if you can stop the promotion of fibroblast lineage cells. And I would assume that it's pretty similarly the case with skeletal muscle as well. So it could be either of those things, probably both.